So this is going to introduce you to some of the consequences of some of those things that Dr. Delcitis was just talking about. Specifically the breakdown of the cells in the tissue. That's a consequence of the wounding that is related to fresh cut processing. So this is a picture of a fresh cut salad that I picked up at the local store in Gainesville, Florida, and just left that on my desk in my office for a day. And this is what it looked like from the bottom, all of that liquid. You see that all leaked out of the fruit tissues, and you can see how cloudy it is also. That's because it's like a microbiological soup there in the bottom of the container. So that's the issue that we're talking about. That's a normal process of senescence in wounded tissue, and it's a real problem for a lot of fresh cut products, especially fruits, including the vegetables that are actually fruit or at least immature fruit. So there's products available to absorb that juice, like in sliced and diced tomatoes here, salads, watermelon. Watermelon is notorious for leaking juice if it's not handled perfectly. There's some peppers. So like I said, this is a wound response in the tissue, and it depends on the extent or the severity of the wounding. So it's directly a consequence of wounding. So you're peeling, abrading, slicing, chopping, shredding, all of these things. So there's the amount of a wounding in relation to the volume of tissue that's involved. There's the force applied, the implement sharpness. So the less force, the sharper the blade, or using a water knife will reduce the injury and reduce the response. So sliced tomatoes are going to have less of this problem than diced tomatoes, for example, because of that reason. And so this is something that's also affected by temperature. So if you do the processing with pre-cooled product and do the processing at a low temperature, that's going to also minimize the wounding effect. This is a slide from Dr. Cantwell, who's in the back there. I don't think you were here during the introduction. She's going to be talking after me later this morning. And this is showing the effect of some of these factors on melon in this case. So these were cut with a cork borer in the lab. It's a metal tube sharpened that used to make holes in corks in the lab, and she used that on the cantaloupe melon. And you can see with the higher temperature and a sharp blade versus a dull blade, you get more of this water-soaking appearance. And the figure on the right is showing electrolyte leakage. And this electrolyte leakage, there are charged particles in the cells, plus and minus charges. And when the cells start to lose their ability to contain their contents and the liquid leaks out, you can measure an electrical current going through the liquid because of these charged particles. So that's electrolyte leakage. It's a way that you can quantify the amount of this kind of damage. So the control depends on minimizing the extent and severity of the wounding that occurs during the processing. It depends on maintaining low temperature during processing and actually through the entire handling system for the products. That's why we recommend 0 to 5 degrees for fresh cut products. Even if there wasn't a food safety factor in all of this, still 0 to 5 degrees is best because it minimizes the consequences of the wounding. And then there can be additional supplemental treatments like adding antioxidants to control browning, and also there's oxidation involved in the damage to the membranes that's related to this leaking. Also firming compounds like calcium to minimize the wounding, the symptoms. Calcium also tends to stabilize the membranes. So you see, for example, sometimes calcium ascorbate. You have the calcium for the firming, you have the ascorbate as an antioxidant. Another way to address this is handling in modified atmosphere packaging. So just like lowering the temperature reduces the rate of these kind of reactions that are going on, if you reduce the oxygen level and in some cases increase the CO2 around the product, that has an additional effect of slowing down these processes, just like the temperature. So just sort of a little aside here in terms of chilling injury. This was something that was considered more of a possible factor early on. Now we don't really think so. If you've heard of chilling injury before, you know that this is a disorder of tropical and subtropical products or fruits and vegetables that originated in the tropics or subtropics. They can be injured by low temperature. And one of the symptoms that's commonly seen from chilling injury is also this translucency or water soaking. But it's been determined that for fresh cuts, there's a time factor involved in chilling injury, and for fresh cuts, because they have such a short shelf life, the time of exposure to the low temperatures is usually not enough to cause chilling injury. So the 0 to 5 is still the best range for fresh cuts, because chilling injury is really not a factor. It's more just general senescence or aging. And so the bottom line is this translucency is due to senescence, not chilling injury. So if you're dealing with any kind of tropical or subtropical product, chilling injury is not something that you need to worry about. You still need to use low temperatures for the fresh cut products. Okay, so consequences of wounding. One of those is induction of ethylene synthesis. It's referred to as wound ethylene. Ethylene is mostly known for its role in promoting ripening of certain fruit. Now, that's why bananas are treated with ethylene, because it stimulates and synchronizes the ripening. Well, post-harvest physiologists like to talk about ripening as a specialized form of senescence. It's just a way that fruit senesce that we happen to like because it involves aroma synthesis and changes in texture and flavor that we like. But it's just another kind of senescence. So you get wound ethylene in the tissue when it's processed. It's greater in fruits than in vegetative tissues. You also at the same time get increased sensitivity to ethylene because of the wounding. It has various effects. It can cause yellowing because it promotes chlorophyll degradation. You get phenolic synthesis stimulated by ethylene, and these phenolics can be involved in oxidative browning because they're the substrate for the reaction. Also, some of the phenolics can impart a bitter taste to the product. You can get tissue softening, obviously, like ripening, but also toughening to some extent, because another phenolic is lignin, which in some specialized cases gets stimulated, its formation gets stimulated by ethylene, and so you can get toughening of the products. Asparagus is the best example of that. And you get this tissue translucency and juice leakage. And so these are all under the umbrella of accelerated senescence, which is promoted by ethylene. And when you wound tissue, it produces greater levels of ethylene, and it just accelerates the whole process that would have happened anyway in the whole product, but it's accelerated in the cut product. So there's a programmed process of death that plant tissues go through. So this is an idealized picture of a cell with the vacuole and the nucleus and the mitochondria and endoplasmic reticulum. And as the tissue or the cells start to die, you get chlorophyll breakdown, you get a slowing down of metabolism, induction of proteases, nucleases, and lipases. These are enzymes that break down proteins, nucleic acid, and lipids. You get cessation of protein synthesis and mitochondrial function. So the whole metabolic system in the cell starts to shut down. And then eventually you get permeabilization of membranes, and the things that are being kept in separate compartments in the cell start leaking back and forth, and you get reactions occurring that aren't supposed to happen or don't normally happen in a healthy cell, and eventually you get cell death. So this is an electron micrograph of a plant cell. So this is more realistic. The cytoplasm is in a small area around the outside, and there's a huge vacuole in the middle. But the thing I want you to notice here is these gas spaces. So there's a cell wall around the cell, and so they're packed together like irregularly shaped bricks being packed together. And at the corners, you get air spaces. So that's actually how gas moves through the tissue in fruits and vegetables for their respiration to proceed. So what happens when all this liquid inside the cells starts to leak to the outside is it fills up these intercellular spaces, and that's why you get the change in appearance and the translucent appearance to the tissue. And so here's an example of pineapples going through their normal process of senescence, and you can see the appearance of those translucent spots that take over greater and greater area in the cross-section of the fruit until you get this translucent appearance, and that's because the cell contents are all leaking out. This is an example of some research that was done with pineapple, looking at modifying the atmosphere as a way to reduce the juice leakage. So this is measuring juice leakage, and there's polypropylene trays and film, there's active MAP and passive MAP, which you'll hear about later today. There's a coating that has a semi-permeable function for controlling gas exchange, and it's just showing how you can reduce the development of the leakage. And actually this was the best treatment, the combination of two things, the polypropylene tray and film and the alginate coating. And here's another example of how this the senescence is related to the translucency. These are fresh cut pineapple prepared from pineapples that were stored for a shorter or longer time. So 2-day-old and 14-day-old actually means days post-harvest, before they were processed. And then this is what they looked like after 12 days at 5 degrees. And you can see that the older fruit had this onset of the water soaking earlier because they were farther along in the process of senescence before they were cut, and then that sped everything up, and they took on that appearance. This is from some work in my lab. This is what happens to fresh cut tomato slices over time. They get this water-soaked appearance. And this is showing the effect of the ripeness stage at which the tomatoes are sliced. So this is what tomatoes cut at either the light red stage or red stage looked like after 8 days at 5 degrees. And you can see that they're much darker, more translucent here. Also notice the missing locular contents. When we picked up these slices out of the containers they were stored in to lay them out for the pictures, the locular gel had just liquefied and just fell out of the slices. So that's what you're seeing here with all these open spaces. This is a picture of some fresh cut zucchini that are way over the hill, and you see more of the same. This was from a graduate student's project looking at machine vision as a way of following the visual changes in a fresh cut product over time. This is some work from my colleague at University of Florida, Don Huber, retired now, but he did a lot of work with fresh cut products. This is showing the effect of exposing fresh cut tissue to ethylene. So these are cucumber slices, and you see on the left how the ethylene exposure has sped up this water soaking. And just sort of strengthening the evidence for the role of ethylene. This is showing the effect of 1-MCP. If you haven't heard of this before, maybe you've heard of Smart Fresh. 1-MCP is a gaseous chemical that binds to the same binding site that ethylene binds to in the plant tissue. And it binds irreversibly, so those binding sites are then blocked and they can't respond to ethylene anymore. So you make the tissue essentially insensitive to ethylene. So if you look at these two bottom pictures here, this is the ethylene exposed cucumber slices, and this is ethylene exposure with 1-MCP. So if you take out the role of ethylene, you inhibit that water soaking. And another one, this is also from Huber and his visiting scientist, Dr. Mao. This is ethylene exposure of watermelon and the effect it has on water soaking. Watermelons are very, very sensitive to ethylene in terms of not only this water soaking, but the tissue will completely disintegrate. If you expose intact watermelons to ethylene, they'll completely liquefy inside so that you can actually shake the fruit and hear it sloshing around inside. Very sensitive to ethylene. And these figures on the side, this is showing the softening of the fruit. So this is the ethylene exposure, the open squares in each case. So that's much accelerated softening, accelerated electrolyte leakage, and this is actually measuring extractable juice. So this is like that juice that collects in the bottom of the containers. And one more, this is papaya exposed to exposed to 1-MCP or not. So here's the control with the water soaking, here's the 1-MCP taking out the ethylene, and that's the effect. You reduce that water soaking. And this is showing electrolyte leakage from those same fresh cut papaya pieces. So this also illustrates the chilling injury point that I was making. This experiment was conducted with whole papayas held at 5 degrees. Obviously, papayas are a tropical fruit. They're not normally stored at 5 degrees because they're subject to chilling injury. This is showing the electrolyte leakage. So these are the fresh cut pieces without 1-MCP, with 1-MCP. And you see over 10 days at 5 degrees, the whole tissue is hardly doing any electrolyte leakage yet, except the control maybe just starting after 10 days, compared to the cut tissue where you get massive electrolyte leakage. So they weren't showing the effects of chilling injury by that time. That's strictly a consequence of the ethylene and the wounding. So another consequence of wounding is membrane lipid degradation. And so what you have going on here is that there are enzymes stimulated that break down the membrane lipids. So phospholipase D is one of those enzymes. It's stimulated by ethylene, like many of these reactions. It results in formation of free fatty acids, which are then the substrate of another enzyme called lipoxygenase, or LOX. The lipoxygenase results in formation of hydroperoxides, which are free radicals. Those are an example of reactive oxygen species, which are also produced in this whole syndrome of senescence and attack the membranes and other parts of the cell. So you get altered membrane protein function, as well as the lipid part starts to get degraded. The proteins that are embedded in the membranes, their environment changes, so their activity changes. You get increased membrane permeability and ion leakage. And then, of course, that results in this water-soaked translucent appearance. Calcium salts can improve membrane stability, and so they reduce that susceptibility to the lipases and reactive oxygen species. So this is showing a couple of these lipases. This is phospholipase C, which doesn't react. This is D that does. This is the intact papaya fruit. This is the cut pieces, and you can see there's an increase in that lipase. And this is showing a massive increase in the lipoxygenase, the other related enzyme. So part of this whole syndrome is also, besides ethylene, you have oxidative stress. And that occurs when the rate of formation of reactive oxygen species, or ROS, is greater than the rate of their removal. So the plant tissue has an antioxidant system that's always around, and it counteracts these reactive oxygen species, which are damaging to not only the membrane but other parts of the cell. But eventually, and especially in cases of wounding and senescence, the ROS overwhelms the antioxidant system. So you get hydroxyl radicals, peroxidized lipids, you get damage to membranes, and in more severe cases, even damage to proteins, damage to nucleic acids, and it helps to accelerate this whole senescent process. And here's where antioxidant solutions, we usually think of something like ascorbic acid being added to inhibit oxidative browning, but it's also helping out in this regard also because it's helping the plant's antioxidant system overcome these reactive oxygen species. This is a figure. This is banana slices or intact banana. It's not really a real fresh cut experiment because they were holding these fruit at 22 degrees C, which is 72 Fahrenheit, so way too warm for fresh cut. It was more of a basic research looking at the effect of wounding on membrane breakdown. Malondialdehyde is a very well-known indicator of membrane peroxidation, so you can see that there's a huge increase in that compound in response to wounding. So to sort of finish up and recap here, how do you reduce water soaking? That's really what we want to know, right? Start with freshly harvested whole products at optimum maturity, right? So the older they are, or the more on their way to senescence they are at the time of cutting, the faster the water soaking is going to appear. So start with the freshest product possible. Pre-cool it and process it at low temperature using very sharp blades. Also, I didn't mention this before, rinse the cut surfaces, but use sanitizer, calcium, antioxidant solutions. Those dips can help reduce the consequences. Package in MAP, which you're going to hear more about later today, and then also maintain the cold chain throughout the handling. So the bottom line is you want to minimize the wounding and keep the temperature low, and that will minimize the consequences of the wounding so that you have less of this problem. I'm going to finish up here. This is something not directly related, but it's interesting in how plants respond to wounding. This is something that was just reported just about a year ago, almost to the day in Science magazine, showing the effect of wounding where there's actually a signal going through the plant. So you don't get the response just at the site of wounding, but it gets transported through the plant. And what these people found was that with wounding, there was an increase in a compound called glutamate, which activated an ion channel. These are pores going from cell to cell, so cells can communicate with each other. And so that pore opened with glutamate binding, and it allowed calcium ions to move from cell to cell at a rate of about 1 millimeter per second, which is like lightning fast for things moving through the plant. And then the calcium they found stimulated jasmonate, which is a sort of obscure compound with hormone properties similar to ethylene that stimulates senescence. And here is a visualization of that. They use genetically modified Arabidopsis that fluoresced upon the binding of calcium. And they're going to, where's my pointer? They're going to cut this leaf right here. And you can see that the leaves that are distant from the point of the wounding, they know that somewhere else in the plant there was wounding that took place within seconds of it happening. So I find that very fascinating. Oh, thank you.